Welcome to the Warnell seminar. This is a particularly bigger crowd. Yeah. Than usual. Very happy about that. Quick reminder for the class people, remember to sign. I'll say it every single time there's someone who forgets about it. So that's why I keep repeating it. And I want to say that I'm reading all your comments and the discussion. They're very nice. I mean, you're getting all the back and forth. You're commenting on speakers. It's pretty cool. So kudos to you all. For those of you who have not put anything yet, go and do it, because the end of the semester will sneak up on you before you realize it. You don't want to, the last day, I need to comment, and your brain is dead by then. So just go now, do it. There's a lot of discussion going on, and yeah, so it's easy now. It's easier now than later. Okay, said that, I will have Dr. Mel introducing our speaker for today.
Thank you. And it's my honor and pleasure to introduce this week's seminar speaker, Dr. Pete Hazelton. I'm gonna give you a little introduction background about Dr. Hazelton. He received his master's actually here at Warnell under the direction of Dr. Gary Grossman. And then he completed his PhD also in Warnell under Dr. Robert Bringoff. So after he completed his doctoral degree, Dr. Hazelton worked for the Massachusetts Division of Fisheries and Wildlife, where he worked his way up and actually became the Chief of Conservation Science, where he did all kinds of different projects related to aquatic conservation, kind of the theme, overall theme of this topic today. Dr. Hazelton came back to Warnell when he was hired as an assistant professor back in 2020, now conducts research under the umbrella of, again, aquatic conservation. One of the focal taxa that Dr. Hazelton works on is freshwater mussels. And again, kind of the overall theme of the topic here today. Dr. Hazelton has a variety of research interests, though, ranging from toxicology to fish to other aquatic inverts like dragonflies. Yeah, it really provides a diverse opportunity for training for students from a variety of different backgrounds, which really is a passion of Dr. Hazelton's. So Dr. Hazelton is well known in the malacology world, frequently collaborates with a variety of state and federal agencies, other university faculty and NGOs. Today, Dr. Hazelton will be providing a synopsis of some of the research that him and his graduate students have been working on over the last few years, again, to gain a better understanding of freshwater mussel biology and ecology. So without further ado, Dr. Hazelton.
I will admit first, but Dr. Hamill will agree that those are the nicest things he's ever said. And number one, I want to thank all of you for coming, especially you fish heads here that are here. It's good to see so many fish heads, but it's good to see a lot of faces I don't recognize. And also, Katerina, thank you so much for inviting me to speak as part of the seminar. And yeah, I'm excited to be here. So probably who I should thank the most is my lab. These are some of the students I've been working with over the past 5 years. Over here, this is, I don't know if my laser will work, on the left-hand side underneath me. These are current students, including 2 co-advised by Jiayun Zhu, and Molly Martin is co-advised by Brian Irwin. And then I've graduated 3 students in the past couple of years. And really part of what I'm going to talk about is the work, obviously, that some of my students are doing, but also about matching opportunities to the right student. So I do have diverse research interests, and so do a lot of the students in the lab. And so we work on a variety of different taxa. Yes, mollusks are pretty high on the list, but we also work on aquatic insects, decapod crustaceans such as freshwater crayfish, and we also work on fishes as well. And in general, a lot of our research is focused sort of around 3 main themes: conservation of biology of native aquatic fauna, the effects of aquatic invasive species, and also the biology of aquatic invasive species, and the effects of contaminants on native biota. So really studying in the ecotoxicology realm as well. So you might ask, if you have such diverse interests, why specifically freshwater mussels? And I'm hoping that will be pretty clear throughout the rest of the talk. Freshwater mussels are a really interesting model organism to study. Number one, they're really important conservation targets. They have amongst the highest imperilment rates of North American fauna, with as high as 70% or more of the species listed as in peril. And 25% of North America's fauna, or the United States fauna, is listed under the Endangered Species Act. They also have really important ecological roles, okay? These animals, they are bivalve filter feeders living in the sediment, and as filter feeders, they're interacting with the water column, feeding in the water column, and depositing nutrients into the sediment. They're critical as nutrient cycling processes, in what we call benthic coupling of bringing nutrients and sediment out of the water column and depositing it into the sediment. And they're also a really important nutrient battery in these systems because they retain a lot of nutrients and other elements. They also provide a lot of structural habitat, which is really important for other macroinvertebrates and fishes. And sort of the last reason from the ecotoxicology side or the realm of my research interest, the sensitivity to contaminants, because they're at this interface of the benthos and the water column, they sort of represent different routes of exposure to contaminants that are both dissolved in the water, but also bound to the sediment that we can't really look at a lot in a lot of other systems. And the young or the early life stages of freshwater mussels are consistently some of the most sensitive organisms in standard ecotoxicology tests to both organic and inorganic contaminants. Probably the most important reason why I love studying freshwater mussels is their complex life cycle. So they have an obligate parasitic larvae stage where it has to attach to a fish. So we have an adult mussel down here. The adult females, we have a gonochoristic species, so females and males are different. The female retains what are called glaucidia, where they larvae. Eventually, those larvae have to make it onto a fish host where they metamorphose over a period of days to weeks, drop into the sediment, and then go on their married life for a pretty long period for an invertebrate, up to 100 years, some of the North American fauna. This whole relationship, this host relationship, is species specific. So some freshwater mussels are very selective in what fish species they use. And this is we'll talk a little bit more about the dynamics of this host relationship a little bit later in the talk. Specifically today, I want to talk about a couple of different projects. Number one, Michael Baker, a PhD student in the lab, has been working on market recapture population estimation in rare mussels to inform conservation. Molly Martin, a master's student in the lab, who's co-advised by Brian Irwin, is evaluating threats of agricultural and municipal chemical stressors to freshwater mussels in the Conasauga River. And then also a recent graduate, Haley Robinson, did some work understanding mussel life history and host relationships in a biodiverse system. So we'll start with some of Michael's work. This chapter of Michael's thesis, or dissertation rather, has sort of come out of some of my work previously at the Massachusetts Division of Fisheries and Wildlife and some of my colleagues, the collaborators in previous grants. The brook floater is the species of interest here. And it was initially listed for or petitioned for federal listing in 2010 as part of the 404 list from the Center for Biological Diversity. In 2016, there was a state wildlife grant that was awarded to 5 different states throughout brook floater's range. And we were tasked with a number of different things. And Massachusetts was the lead state at the time, and so I was the technical lead on the project. And we were tasked with creating a working group that could identify research needs and also coordinate conservation approaches throughout the whole range. And on the right here, you can see this is the global range of brook floater from the maritime regions in Canada all the way down to the Savannah River in tributaries of the Savannah River here in Georgia and South Carolina. A couple of our other tasks on the grant were to develop protocols for monitoring throughout the range. So to develop protocols that could be used consistently in different regions or different states, because there's not a lot of uniformity or standardization in these types of protocols. So we're often comparing apples to oranges in monitoring freshwater mussels throughout time. And in particular, we developed a protocol that was focused towards market recapture monitoring of this species. And these are some of the data. This all started back in 2018 and were sampled. There were 5 sites that were sampled, 3 in Massachusetts and 2 in Maine. And we sampled these sites back in 2018 to 2020. And so this all happened before I even came to UVA, let alone Michael. The sites ranged from around 30 to 120 meter lengths reaches. And we use a lot of visual and tactile search methods for freshwater mussels. So essentially, we're in a stream, snorkeling, looking for them and using our hands to find them. So as you can imagine, our detection of mussels is actually pretty low compared to a lot of other species, because we have to rely just on our visual. We don't have any gear. We have to rely on our eyes in water and also what we can feel. And then each site, according to the protocol, each site was visited a minimum of 2 times each summer. So in comes Michael for part of his dissertation, and he was able to use robust design marker recapture methods to evaluate these populations in Maine and Massachusetts. And a couple of functions about robust design methods is they combine open and closed models, or combine open and closed periods during which you can estimate things like, so these you have 3 primary periods, which are open, and those would be, in this case, years. Our primary periods would be year 1 and 2. And then we have secondary periods or sampling occasions. So within each year, we would have 1 or 2 secondary sampling occasions. And so we actually had 3 primary occasions, years 1, 2, and 3. And then we just used 2 secondary sampling periods within each year. And so the secondary sampling period is a time in which the population is assumed closed, so there's no immigration or immigration occurring. And this is the minimum data set that would be required for this type of model. But it's helpful because it provides estimates of 3 different parameters: survival, capture probability, and also abundance, or population size. And so this is, I will admit that there are many professors here at Warnell and elsewhere that are far more better read than I am in these population models. So I'm just going to glance through some of the things that we actually found to get to some of the meat of Michael's project. So first is survival. We come up with estimates of survival between these different populations. Massachusetts is over here in white. Maine is over here in dark gray, and survival is pretty high in all of them, right around 80 to 90% in every single population. So greater variability in these Massachusetts populations. The next thing we can get from these models is capture probability. So the probability that we're going to capture any one individual or recapture it. And what we see is in Maine, there's a pretty high capture probability, probably around 70 to 75%, whereas in Massachusetts, it's quite variable, but we're actually down around 0.3. And this is probably from some of the other studies, 0.3 is pretty consistent with freshwater mussel work. And there may be, we can go on and think about different reasons why there's differences between these 2 populations, but for the purposes of the work here or my talk, it's not really that important. From those other parameters, we can get an estimate of abundance. And so here we knew ahead of time that the populations in Maine were going to be much larger than those in Massachusetts. And so we have these populations over here in Maine, which are quite large, on the order of 200 to 300 animals, or 150 to 200 animals. And then these populations in Massachusetts were quite small, around 50 or fewer animals, or upwards around 100. What you'll notice is the error rates between these different populations. So they're quite large. Those confidence intervals are quite large in the Massachusetts population. It's probably driven by the variability in the low capture probability. So we're going to forget about Maine for a minute. And we're going to think about this. How does this apply to somebody who's a manager for monitoring? And so we're just going to take the Farmington River and presume for a minute that we actually go back 5 years later and survey 3 years again. and we find a 25% decline. Presumably you could do the same thing in 2028, and that's 2030, not 200 and 230. And we see a 75% decline. Given those confidence intervals, can we be certain that that decline is occurring? Anybody? No, we can't be certain that those declines are occurring, okay? So if we looked at the average population size across these 3 years, these 3 years, and these 3 years, our confidence intervals are quite large. And so we may not be able to actually determine whether there's a difference in the population size. And so if we were to pose this as a hypothesis, whether for monitoring for conservation purposes, we could say that the null hypothesis is that populations are remaining the same through time. And our alternative is that the populations are declining through time. And we'd have to evaluate that statistically. So there's a couple things we have to think about with evaluations like that. With statistics, we're also often concerned about type 1 error, which is a false positive. That's when you accept the alternative hypothesis when the null is true. And we use alpha of 0.05 to control for this, right? To a manager, what this might mean is thinking a decline is happening when it's really not. There's also type 2 error, which is a false negative, when we reject the alternative hypothesis when it's actually true. And so this is associated with beta, and we often use power, or 1 minus beta, to evaluate what our ability to detect false negatives, right? If we're to put this in the perspective of a manager, this is not realizing a decline is occurring when it actually is. Which is more important to a conservation manager? Type 1 or type 2 error?
Type 2 would be worse.
Type 2 would be worse, right? Not being able to detect a decline is occurring when it actually is a critical scenario for a manager. We want to know that we have enough data to be able to determine that a decline is actually happening. We don't want to assume it's not happening when it is. So what Michael has been really interested in working on is simulating capture histories for using or conducting a power analysis from simulated data. And so he's simulated capture histories for all the animals in a population. And I'm going to walk you through how he's done this. He's done this many times for several different populations, and the populations have varied in size, 50 to 300 animals. He's varied the number of secondary periods or secondary samples that he's sampled that population. And then there are some parameters that we've actually kept static. So the number of primary periods or years, the yearly survival of each individual, and then the capture probability of 0.3, really similar to what we found in those Massachusetts animals, right? And so he's taken those scenarios and populations, and he's simulated that scenario 1,000 times each, all right? And he's then calculated using robust design approach to estimate abundance using those capture histories. And so what we have on the left is a population of only 50 animals. On the right is a population of 200 animals. And on the bottom, the horizontal axis is the decline in population, whereas power is on the vertical axis. Looking at only 2 secondary periods in the dark black, then we go to 3, 4, and 5 secondary periods. So that is the number of times you sample within a season. At a small population of only 50 animals, we only have 50% power to detect a 60% decline in a population with a population that small. A coin flip to detect whether the population is declining by 60%. Okay? If we move that up to 5 sampling events in a given year, we can bring that down to around 25% decline in population. So it's still a coin flip, but we might be able to detect a 25% decline in the population. It gets a little bit less dismal as we look at a larger population size. So it's typically right around 30% for a 2 sampling event, secondary period. When we get up to 5, it's probably closer to a 10% decline we can detect if we're using the appropriate or five secondary samples. So all of this is to say that the number of times you sample is really important. A lot of these methods that we've been pushing out to managers, and there's not, they may be undersampling what they need to identify a decline in population. So the power to detect population declines with the number of the power to detect a percent population decline with increasing the number of secondary sampling periods will actually increase your power to detect population decline. And specifically, this is more important when the population is very small. We may never actually get to a point in a very small population where we can be confident that we're detecting a population decline that's smaller than about 20%. So one of the other projects we've been working on, Molly Martin, is assessing the contaminants to freshwater mussels in the Conasauga River. The Conasauga River up in Northwest Georgia has a high rate of species diversity. We've got around 80 different fish species, crayfishes around 10 species, and 28 mussel species. And the fishes and mussels in the area are experiencing what some people call enigmatic declines. We can't really pinpoint what is causing declines of the fauna, both populations, but also species in the area. And the watershed has high mussel imperilment rates. There's 7 species listed under the ESA and 4 species listed under the Georgia Endangered Species List. Some of the threats we know are potential land uses or associated with potential land uses. The upper tributaries of the river are actually pretty well forested and protected in the Chattahoochee National Forest and also the Cherokee National Forest. But as you get down into the lower tributaries, there's a lot of agriculture in this yellow, and then also a lot more urban and municipal development in these red areas. So Molly wanted to identify what the contaminants might actually be, if there are contaminants that are driving this associated with both agriculture and municipal development. And so she evaluated sediment contaminants in the lab. It's a 2-part study, and then is also evaluating in situ exposures of water quality in the field. We've got about 13 different sites spread out throughout the watershed, and we've listed these as impacted sites in the yellow triangles, which have less than 75% forested land cover, and blue is unimpacted. And so really what's more important here is understanding those 2 colors of blue and yellow. And so some of our parameters of interest, our response variables are, we looked at survival. We're not going to talk about survival because we didn't find a difference in survival in any of our analyses, but we looked at change in length, percent change in length, and percent change in biomass as measures of growth in both of these tests. For the sediment test, we've got about 29 different explanatory variables and about 43 different explanatory variables, and these range from landscape parameters, sediment chemistry, water chemistry, and then also in the in situ tests, temporal variations, because we measured monthly mussel growth throughout a 5-month season. So I'll talk a little bit about the, and all of this is to say that it's a pretty complex database with only 13 sites, and you have up to 43 potential explanatory variables. So the data that I'm going to present is preliminary, and we've got a lot of data reduction and analysis ahead of us, but some of these data are being analyzed right now. So the sediment toxicity trial, how this works is we take a bunch of 30 to 60-day-old mussels. We put them in these beakers with sediments that we collected out in the wild. And we grow them out using the same water over a period of about 1 month, and then we measure their growth. So what we have here is the change in length for the sediment toxicity trial. Percent change in length is on the vertical axis, and then our different sites are on the horizontal axis, with yellow being those impacted sites and blue being those forested, heavily forested and unimpacted sites. So we're definitely seeing a difference across sites. This red dotted line is the mean percent change in growth throughout the 1 month trial. And one of the things that we keep coming up with when we're running preliminary models is that magnesium is really important. And so we're seeing a significant reduction in percent change in growth or percent change in length with increasing values of magnesium within the sediments. And this is sort of supporting what we have seen in the literature, not necessarily in sediment, but in water-only exposures, that magnesium is potentially toxic to freshwater mussels and it reduces growth and other sublethal measures. So those silo studies, this is a little bit different. These, instead of the sediment, we expose these animals in situ. So the animals are placed into concrete silos, which looks like a giant concrete salad bowl, but it's got a chamber inside of it. And as water flows over the top of this chamber, water underneath is able to flow through. So we're exposing the mussels directly to the water and anything that's in the water, rather than exposing them to things that are in sediments that we collected in the field. And so these were done over 5 months, and we were able to measure mussel growth every month, as well as water quality parameters. And this is a similar graph showing the percent change in length and also the stream name. Same color difference between unimpacted and impacted sites. And we're seeing some sites are quite different in the percent change in length, but we're not seeing a lot of consistency in unimpacted or impacted sites. What's really interesting to us is in these early models that we're running, we're finding that there's a quadratic relationship with chlorophyll. And so chlorophyll is measured from its the photopigment that is measured as a indicator of algae. Algae is something that mussels eat, as well as bacteria and other things. But similar to other studies, we are seeing that there's an increase in growth at the lower end of chlorophyll, but there might actually be sort of a tipping point. And some sites, although it's only one data point down here, we're seeing that there may actually be too much algae and mussels are actually not growing as well. There's still a lot of work that we need to look at with other contaminants potentially. We have about 13 pesticides in this study that we haven't analyzed yet. And so there's still a lot left to do, but this is something, this growth, seeing that growth is controlled more by algae than contaminants is something that's been seen in other systems as well. So again, overall findings here, still quite preliminary, but in the sedimentary trial, we're looking at magnesium, maybe reducing growth. The silo trial, really the quadratic relationship with chlorophyll A is pretty interesting to us. But what's interesting to us is that when we compare these two things, we've got the sediment trial on the top and the silo trial on the bottom. One of our impacted sites, Mill 3, this is actually right below the city of Dalton. We're seeing vastly reduced growth in those juveniles that are treated in the sediment trial. However, we're seeing increased growth with those older animals. And so these are two different exposures. One set is being exposed just to the sediments, one set is being exposed just to the water column. And so there may be inference here for the exposure route of any given contaminant, but also the timing of exposure to individual mussels as well. So we're going to have to unravel this quite a bit more or work on it a little bit more, and much to Molly's dismay. And then finally, if there's something that really, I love conservation, I love ecotoxicology, these questions are really fascinating to me. but understanding the mussel life history is really an amazing, amazing process. So this is a project with Haley Robinson did her master's on this, and John Wares is here as a major collaborator on this project. And there are multiple groups that were incorporated that assisted either with funding or other sources. And this actually came from a grant from the Georgia Water Planning and Policy Center, and we collaborated with Albany State University and also the Flint River Aquarium down in Albany, which is a public aquarium down in Albany. And I'm happy to say that this has been accepted from Freshwater Science and will be published this June. A little bit about the history of the Flint. This is the Flint River down here. Runs from under the Atlanta airport all the way down to where it joins the Chattahoochee and Lake Seminole, and then out through Apalachicola Bay. There's 28 species of freshwater mussel in the Flint. 5 are federally listed, and 4 are listed under Georgia Endangered Species Act. We don't know the host fish. I said earlier, there's a species-specific relationship with a lot of these fishes and mussels. And we don't know the host for 40% of the mussels that are in the Flint River. So herein is part of our research question, is to understand a little bit more about the population of mussels or the community of mussels and fish in the Flint River. Give you a little bit more detailed look. As I said earlier, male and female mussels are different. Males release sperm packets into the water. They're spermatozoogmata, which actually swim in little balls around the water. Female mussels filter those up, and she fertilizes her eggs, which develop into glaucidia in her gills. Okay, somehow those glaucidia have to get onto the fish, right? They get onto a fish's gills or fins, and they become encapsulated, and they metamorphose over the period of days to weeks, and then eventually transform into these small juveniles, on average only about 1/4 of a millimeter large. Okay, they eventually excise from the fish, fall down to the sediment, grow into juveniles, and we might not see them for another 2 to 3 years until they're large enough to be sampled. And again, these have quite high longevity. There's some animals living decades to even 70 to 90 years, 100 years. How do they get on the fish? How many people see 3 fish in this picture? How many people see 4 fish in this picture? How many people see 8 fish in this picture? Kelly, there's the door. So there's only 3 fish in this picture, all right? Here's 1. These are darters. Here's 2. Here's 3. Sorry, my light is not very good. 3 is up in the corner. Some of you are asking, what's this dang thing, right?
Yeah.
So underneath this is a freshwater mussel. Okay? This is a lure. That's part of its mantle. That is a mimic, an aggressive mimic of these fish to lure bass in to attack the mussel so it can release the glaucidia onto the bass. This is what it looks like when it's actually moving. It will flap this lure, and underneath it are its gills that are full of hundreds of thousands of glaucidia or larvae that will latch on to a fish's gills and fins as soon as it hits it. Okay? They don't all look like that. This is a video that I've taken of a mussel called an eastern pond mussel. It doesn't look necessarily like a fish, but it's just radiating or undulating its mantle so that you can see the white gills underneath. I know that some of the faculty in the room remember the TV show Knight Rider. This is very reminiscent of this. This is the Knight Rider mussel. This is the sound. Remember? It's like something like that. There's also external lures. So some of these mussels can create mimics that are extended on long strands of mucus that are packed full of these glaucidia that might look like a larval fish, might look like a worm. And even here we have a mimic of a black fly pupa. Okay, so this is what's called a supraconglutinet. That's the mussel lure. And it has an eye spot, segmentation of the abdomen, gills. This is a drawing of a black fly pupa. Okay, so this is what's called aggressive mimicry, very similar to what an anglerfish would use to lure a host in and infect it with its larvae. And there's all sorts of other attraction strategies that mussels use, from these external lures, large lures, and they're related to different fish species. So we've got carnivorous fish attacking those mantle lures and external lures, all the way down to we have broadcasters, which are generally host generalists, and even some mussels that use trapping of fish, or even maternal sacrifice, where the mussel basically wants to be eaten by a freshwater drum to make sure it infects the fish. How we identify these host-fish relationships. In the lab, traditional mussel hosts, we would bring in a mussel, a gravid female, and bring in a bunch of fish. We collect the Glaucidia, put them on the fish in these tanks, and then we collect the Glaucidia and juveniles that fall off at the end. Hopefully there's juveniles, and we kind of identify, well, that was a successful host. This host is physiologically suitable to be a host. There's pros and cons to this approach. Number one is that it's for the pros, is that it's really efficient to screen for physiological compatibility. The con is that it ignores all ecological interaction between the host and the mussel. And it might identify unlikely hosts. A fish might be a physiologically suitable host, but may never interact with a mussel, with that species of mussel in the wild, right? So how do we get around this? Well, we wanted to look at this in a different way. We wanted to identify wild host relationships in the lower Flint River. And this is only something that's been done in a handful of studies. And so the idea here is we can collect a species, in this case, it is mussels that are attached to fish by going out and collecting fish. And then we can collect the mussels as they fall off the fish, collect their DNA, analyze their DNA, and use barcoding analysis to identify what species of mussel it is. All right. We looked across 9 different sites that were based on Georgia DNR mussel data, and fish were collected, and typically in May. Most of the study was done in June in either 2021 and 2022. And we collected a representative fish sample from the field. So a good proportion of all the different species that we found, and we returned those either to the Flint River Aquarium or to the University of Georgia. And so part of why we spent them to the Flint River Aquarium is I mentioned earlier that part of this study, one of our partners was Albany State University, which happens to be an HBCU. And they have a really awesome bench biology program. They have some great equipment on molecular biology, cellular biology. They have a really fantastic faculty there. They don't have a lot of experience in field or natural resources. So this whole grant was a training grant to Albany State and the Georgia Water and Policy Center, where they hired us to do the science, but also training students from Albany State in these natural resources. So the students were out collecting fish with us, with the backpack shopper, identifying these fish. They were conducting the animal husbandry, where we brought the fish in and had fish in tanks and mussels were falling off. This is actually happening inside the gift shop of the Albany River Aquarium, right behind a glass wall, and we had posters there. So as people were coming through to the aquarium, they were seeing a poster about the life cycle of a mussel, and they were seeing students working right there in the river aquarium on science. Okay, not only connecting them to their research, to the resources of the Flint River Aquarium, but on science. So students were looking through microscopes on a bench right behind this tank system. So this is Rodness. He's looking through the microscope there. This is our first year, and he's he's identifying whether we have an untransformed glaucidia, which just looks kind of like a castanet. It's folded open right now, or a transformed juvenile, which has some tissue differentiation inside of it, and it's closed. Okay, so we can identify whether this is untransformed, it's just a larvae, or it's a juvenile. If it's a juvenile, we know that is confirmed that that fish is a host for this mussel. The students were also responsible, and all of these activities were also happening here at UGA, that my grad student Haley was conducting. So the students were responsible for DNA extraction, polymerase chain reaction and running gels. And then when we find positive hits, we would actually take those samples and we would send them out for sequencing. DNA isolates were sequenced for the COX-1 gene, which is pretty conserved across all the many different taxa. And we compared these with a library of known mussel species sequences on GenBank using BLAST, or the basic local alignment search. And we used 97% pairwise identity as an identification threshold. What that means is that we have, you can think of all of your base pairs on a mussel gene here, but it's 658 base pairs long. And so here's a sequence that is from the database, all of these sequences here, and we have to have 90% or more match between a species on the database and our sample for this to be a positive identification, okay? And in this case, this one actually out of, what is that, 10, 11 base pairs, something like that. We've got a couple of mismatches, so this wouldn't actually fit. So that sample is not a match with that species. What we found amazed us. Out of 529 total fish over the 2 years, we managed to have them in 320 tanks between the river aquarium and here in UGA. 829 mussels dropped off, and 58% of those were juveniles. This is far more than what we would be able to identify using a traditional host fish project or approach. And so we've got, well, just to show you briefly, I won't go through all the different fish species here, but these are the different drop-offs. So some fish species just had more mussels, and this is total, not average. So we have some things like Lepomis punctatus, which is the spotted sunfish. We had many, many more juveniles and more glaucidia fall off those than most other mussels. It's also the fish we caught. What's most important is these relationships, okay? So we have mussel species over on the right-hand side, and we are able to identify 5 different types of mussels compared to fish on the left-hand side. And why I say types of mussels is certainly down here we have 4 different species, below the vibex, Utterbachia bacillus, toxoplasma, and Leonio leonosus. And then that big gray group of Elliptiospa is because that is a genus where we can't actually get 97% difference between species. We just can't genetically tell the species apart. So that may actually, however, 50% of the mussel species in the basin are Elliptios. Maybe not 50%. That's pretty high. The most common genus in the basin by far. So that may actually represent about 5 different species. Some general trends that we're seeing. Utterbachia and bacillus down here, we know that this is a host generalist. We only found 2 different mussels, and it's actually on a darter, which is in the perch family, and then Pteranotropus, which is in the minnow family. So these are very distantly related fish, not surprising from a generalist. These other 3 species, Leonio, Toxoplasma, and Velosa, are somewhat related, and all 3 of them use external, or not external, but use mantle lures. Velosa vibex has 2 positive hits on Essex niger and Essex americanus. Those are pickerel, a visual predator. And so this is a mussel that's using an external lure, or a mantle lure, sorry, that is bringing in a visual predator. Toxoplasma palmum, same thing. We have positive hit on Lepomis, which is a sunfish, actually 3 different Lepomis species. And then Leah Unio, again, uses mantle lures. We have 2 hits here with stars, which means that there were Glaucidia only. We can't confirm that that's a physiological host. But also, again, with Lepomis, 2 different sunfish species. So all 3 of those connections are with a mussel that has a lure and a visual predator, fish predator. Some of the overall findings, we have 36 unique mussel and host relationships, and almost all of those were novel. Okay, meaning that they've never been reported from traditional host trials at all in the literature or in databases that we're aware of. 19 of the 30 fish species that we tested hosted a mussel. And the relationships, we also, as a caveat, this is only from June across 2 years. It's very, very possible and very likely. We know mussel phenology can vary. So this could be applied many, many more times, and we could actually start to map out what the host fish relationship is. And so future directions, John and I are really interested in increasing the genetic library for further mussel identification. And then also another student is evaluating Glaucidia and juvenile morphology to identify mussels. So Amy's taking we have pictures from all of these Glaucidia and juveniles that fell off of all of our mussels. And she's using morphological assessment to be able to map that back to these different species of mussels. Maybe we can use morphology as another. Some of the student learning outcomes with Albany State University, I can't understate how important this is for me, is we trained 11 interns from Albany State on things that they never got experience in 4 years of their biological education there. So they were trained in research process, they were trained in fish collection techniques, aquatic animal husbandry, basic light microscopy, DNA extraction and PCR. Those 2, they actually had a little bit of experience. Some of them had a little bit of experience, but then also barcoding analysis using BLAST. So the whole project was also done as a workshop and a training opportunity for these students. And this has kind of been a trend throughout a lot of my time in mentoring since I've been here. And I've had a great opportunity to mentor some other folks or provide opportunities for other folks. Maisha Epps was a visiting scholar from the NIH Maximizing Access to Research Careers in the summer of 2022. And she worked on Haley's project, the host fish project. And then Kendra Collier was an intern on the Hutton Junior Fisheries Program intern with American Fisheries Society in the summer of 2023. Actually, that's incorrect. It's 2024. It's just last year. And helped out on Molly's project a lot. So I'm sort of showing this to say that while I said earlier, providing opportunities to my grad students is really important to me, but mentoring happens on many different levels. And so I've also had the opportunity to mentor several different technicians here in Warnell and across UGA. And I've been really proud to be able to work with all of these students. And with that, I want to definitely thank them because the work that happens in my lab isn't possible without my students, obviously. And with that, I will also like to thank you, and I'm happy to answer any questions that you have.